All right, good morning. We are doing our graphic organizer notes for our Module 7 Biotechnology unit. So if you will go ahead and turn in your handbook to page 7A. Looks like this, to page 7A. All right, so the first thing in your notes that you read through was all about DNA fingerprinting. And DNA fingerprints are basically created using a GES or gel electrophoresis system. And the steps involved in that are that the restriction enzyme cuts or cleaves the DNA. So you'll see the letter C to represent either cut or cleave. So it's cutting it like a pair of scissors. All right, then that DNA sample is then loaded into the gel electrophoresis system or GES, and that electrophoresis sends electrical currents that then separates out those DNA fragments. And those DNA fragments are separated out by length. All right, so your shortest fragments are going to travel the furthest, and your longest ones are going to stay kind of more towards the very start of the gel system. So this banding pattern that's created is then compared to other samples. So in your graphic organizer, you can see right here, we've got an example of a gel electrophoresis, and that banding pattern is showing us the mother, the child, and then a potential father for that particular set of DNA. All right, and you can go in and you can actually look at and see whether your bars line up. And remember that a child receives 50% of their DNA from mom and 50% from dad. So each band on the child should match up with either the mother or the father. If it does not, then that means that the father is not that particular child's parent. All right, so looking at this particular sample, and this is the one that's on your graphic organizer on page 7A. All right, we see our mother, our father, and our child. So the first bar for the child, notice that it matches up with the mother, so that's a check plus for that one. The second bar for the child then matches up with the father. The third bar for the child, notice that it's in the same line as the mother's bar, so that works. The fourth bar matches up with the father, and the fifth bar matches up with both mom and dad. All right, so for this particular instance, this father is potentially the father of this child because all of the child's DNA lines up with either mom or dad. And this DNA fingerprinting, basically, if you ever watch what I refer to as trash TV or that Maury show, and they say, you are not the father, it's because they've gone through and they've done this DNA testing, and it can help to determine paternity. It can help to aid with forensic science. So when we're looking at crime scenes and blood that's left behind by either a victim or the perpetrator themselves. And it's used for cataloging endangered species as well. All right, so the next thing you're going to fill in on your graphic organizer is where it says at the top that genetic engineering is modifying DNA. So it's highlighted in bright yellow here. At the very top of the right-hand side, it says genetic engineering, and it has a line right down modifying DNA. And this genetic engineering can be used for creating GMOs, where it says GMOs, aka that means GMO stands for genetically modified organism. All right, these GMOs can actually be good and they can be bad at the same time. So GMOs can be used in medicine. We use them for gene cloning. So basically what we'll do is we'll take a vector, which is a bacterial plasmid. So remember that we talked about plasmids were circular rings of DNA found in bacteria. And what they'll do is they'll cut open that plasmid. They'll put in a section of DNA from something else. So say a human DNA for the gene for insulin production. And what they'll do is they'll put it into the DNA, the bacterial plasmid. It'll form something called recombinant DNA, which just means recombined DNA. And then that DNA is then put back into the bacterial cell. And we talked about before that bacteria goes through asexual reproduction. And asexual reproduction means that bacteria is basically just splitting, like you're taking a ball of Play-Doh, splitting it in half, taking those two balls, splitting it in half. So it happens really, really quickly. And then that bacteria is actually producing human insulin, which is kind of neat if you really think about it. So this was a huge step in medical science when it came to studying diabetes and the production of insulin, because originally the insulin was taken from pigs, but that is kind of a hard thing to do when so many people need it. So now the bacteria can actually produce human insulin. All right, GMOs can also be used in a couple other things. They can be used in agriculture. So where it says agriculture, an example, I want you to write down that it can produce corn that is actually pest resistant. So instead of putting all these harmful pesticides on our crops, scientists have actually genetically engineered corn crops. Corn can actually be genetically pest resistant, and so scientists and agriculture and farmers don't actually have to put pesticides on our crops anymore. GMOs can also be used in industry. So there has been bacteria that has been genetically modified to actually clean up oil spills. So when we had the big giant Exxon Valdez oil spill, scientists actually started creating bacteria that would eat the oil in the oceans, and therefore kind of clean it up themselves in a more eco-friendly manner than going in and trying to scoop out all of this oil before it started to harm our ecosystem. All right, so let's talk a little bit about gene therapy as well. So down over to the right-hand side, it says gene therapy, and it gives you the definition. I want you to copy down what's highlighted in yellow on the screen here. All right, and gene therapy basically just is replacing a defective gene with a functioning gene that then serves as a vector, such as a virus. All right, so scientists will actually take viruses, they will replace a defective gene with a functioning gene in that virus, and then that virus will reproduce over and over again, and it will actually modify the DNA for that individual. All right, so our steps that we take for our gene therapy, and copy these down where it says 1, 2, and 3 under steps, is to identify the defective gene to begin with. All right, so some of the diseases that can actually be cured using gene therapy are things like cystic fibrosis, which is shortened to CF a lot of times, and then SCIDs. All right, so in gene therapy, we take that defective gene, we basically package a functioning gene into that vector. So we take a good gene, take out the bad gene, put the good gene in, and then introduce that vector to the organism. So they'll take out the bad gene, they'll put in a good gene, they'll put it in the virus, and then introduce that virus to that particular individual. And when we talk about viruses as a vector, we are not saying something that will be harmful to that individual. This is something that is a good thing. Gene therapy is a positive for that person. All right. The Human Genome Project was a huge scientific step. And basically, the Human Genome Project, or HGP, was when scientists identified the location and the sequence of the genes on the human chromosomes. So it was basically kind of this race in order to map out human chromosomes. So it allows scientists and medical professionals to actually know where the defective genes are and then locate them in order to use that gene therapy. All right, and then the last little bit we're going to talk about is something called stem cell research. And a stem cell is basically just defined as an unspecialized cell that can have the ability to differentiate, so it can turn into and it can become one or more types of specialized cells. So a single stem cell can actually differentiate into things like muscle cells, intestinal cells, liver cells, cardiac cells, nerve cells, blood cells. It can become basically anything that it wants to be. So think of it kind of like a like a blank canvas. It can differentiate into all these different types of cells. All right, so there are two different types of stem cells, embryonic stem cells and adult stem cells. And embryonic stem cells are actually taken from eggs that were fertilized in vitro, meaning that they were what we refer to as kind of a petri dish. So this in vitro fertilization means that the egg and the sperm were joined outside of the body in a lab and then implanted into the individual. All right, so these particular embryonic stem cells can differentiate into many, many, many different types of cells. They are the ultimate blank canvas, whereas an adult stem cell is found in adult tissues, and they're actually more limited into the types of cells that they can become. But this reason for all of this different stem cell research, we can use it to treat cancer, we can use it to test drugs, we can use it as replacement cells for diseases, things such as Alzheimer's or spinal cord injuries. And that is why stem cell research is so incredibly important. This whole idea of biotechnology and all these different things that we can do with our genes and our cells has opened so many doors to medical advances.